Okay, so ladies and gentlemen, you are welcome. So in today's lecture, we are going to look at MCQ in chemistry of amino acid and proteins. So I'm going to project or share the MCQ slides, and I will be reading the questions, and then you will be I will be asking you the questions and you will be answering, and when there is any mistake, then I will actually correct it. So let me start by sharing the slides. So you can write on the comment section. So can you hear me? Write on the comment section. Can you hear me? Please, can you hear me? So you are all welcome. So I think you can write on the comment section. So let me start actually going through the questions. So I'll be asking you. So if you know the answer, you've actually put it on the comment section. Please, can you hear me? Can you hear me? If yes, please write on the chat box. I said if you can hear me, write on the chat box. So the first question says, which is question 1, the chirality of an amino acid results from the fact that it is alpha carbon. Option A has no net charge. B, it is a carboxylic acid. C is bonded to 4 different chemical groups. C is in the L absolute configuration in naturally occurring proteins. So ladies and gentlemen, what is the answer? Okay, Jasmine, good evening. So this is the questions. The chirality of an amino acid result from the fact that it is alpha carbon. So A, question A says has no net charge. B is a carboxylic group. C is bonded to 4 different chemical groups. And then D is in the L absolute configuration in naturally occurring proteins. So ladies and gentlemen, remember in actually amino acids, we have 20 different amino acids, and 19 of the amino acid has chiral carbon. So what make the amino acid to have that alpha carbon? Option A says has no net charge. Because it has no net charge, that is why it has alpha carbon. Option B says is a carboxylic acid, that is why it is alpha carbon. Then C is bonded to 4 different chemical groups, that is why it is alpha carbon. And then option D says it is in the L absolute configuration in the naturally occurring proteins. So what is the answer to this question? If you know the answer, put it on the comment section. What is the answer to this question? So I want actually the discussion to be more of interaction between me and you. So I will be actually reading the questions, then I will be asking you. So if you know the answer, just write the answer on the comment section. So if it is correct, then I will answer you as correct. And if it is wrong, I will give you the correct answer. So please, what is the answer to this? I can see that it's Rahama Ismail, Mimi, There is Banyo. So what is the answer to this question? For this, you should be very fast while writing on the comment section. What is the answer to this question? Okay, Raquel, that is Anzaku, says the answer is C. Yes, correct. The answer is C. So all the amino acid having four different groups attached to it, that amino acid is described as alpha carbon. And we have 20 amino acids that have alpha carbon. We have 19 amino acids that have alpha carbon. So which of the amino acid that doesn't have alpha carbon and why? Which of the amino acid out of the 20 amino acid doesn't have alpha carbon and why? I'm waiting for your response. So I think that actually I realized that it's like you are delaying in writing the answer. So in reply, okay, so Idris Abdurrashid says it's glycine. Yes, the answer is glycine. So glycine is the amino acid, is the only amino acid that doesn't have alpha carbon because it has two hydrogen atoms attached to it. So now let's move to the to another. No, it's not proline sule. The amino acid that doesn't have alpha carbon is glycine. So as we can see clearly, the answer to this question is option C, where the carbon is bonded to four different chemical groups. So then let's move to another question. Then of the 20 standard amino acids, only dash 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 is not optically active. The reason is that it is side chain A, the A, the amino acid that is not optically active, alanine, is a simple methyl group. So because the side change is a simple alanine with a side change of simple methyl group, B, glycine with a side change as a hydrogen atom. Glycine is unbranched. B, lysine contains only nitrogen, and D, proline forms a covalent bond with the amino group. So what is the answer to this question? So which of the amino acid that is not optically active? So we have the option here. We have alanine, we have glycine, we have glycine. Then the side change is a hydrogen atom. Then we have glycine. The side change as unbranched. We have lysine, contains only hydrogen. And then we have proline, forms a covalent bond with amino group. So what is the answer to this question? So I think, ladies and gentlemen, without wasting much of our time, we can see that glycine is not optically active because the alpha carbon or the central carbon contain contain two hydrogen atoms. So it's not actually four different groups that attach to it, and that is why glycine is considered as not optically active amino acid because it doesn't have or it doesn't contain chiral carbon because the side chain contain hydrogen atoms, making it to have two hydrogen atoms attached to the central carbon. So then the answer to this, of course, is option B. The answer is option B, so glycine with a side change of hydrogen atom. So let's look at question 3. Two amino acid of the standard 20 contain sulfur atoms. They are, so we have some of the amino acid that have sulfur in them out of the 20. So A, we have cysteine and serine. B, cysteine and threonine. C, methionine and cysteine. D, methionine and serine. And E, threonine and serine. So what is the answer to this? Which of the amino acid, which of the following amino acid contains sulfur in them? Which of the two amino acid here contains sulfur in them? So what is the answer? A, cysteine and serine. B, cysteine and threonine. C, methionine and cysteine. D, methionine and serine. And E, threonine and serine. So what is the answer to this question? What is the answer to this question? Which of the two amino acids that contain methionine and serine? Okay, Jasmine says B. No, B is not the answer for this question. So question 3, the correct answer is what? So for question 3, the answer is methionine and serine. The answer is methionine and serine. So option C here is the answer. Option C here is the answer. So we have methionine and cysteine. So let's move to question 4. So all of the amino acids that are found in protein except for proline contain AAA dash. They contain all of them. They contain amino group. B, carboxylic group. Carbonyl group. C, carboxyl group. D, ester group. E, thiol group. So the answer is already written as an amino group. So all the amino acid that we have, they contain all of the amino acids that are found in protein except proline contain amino group. So it's only proline that doesn't contain amino group. So the answer is option E. So all of the amino acid except proline contain amino group. So then let's move to question 5. Amino acids are ampholyte because they can function as either A, acid or a base. B, neutral molecules or an anion. C, polar or non-polar molecules. D, standard or a non-standard monomer in proteins. E, transparent or a light absorbing compound. So why amino acid is considered as an ampholyte? So when we said amino acid is an ampholyte, is because the amino acid contain both carboxylic group and amino group, and that is what make it to have both acidic nature and a basic nature, and that is why it's considered as an ampholyte. It means it has duality, or it has dual properties in terms of acidity and basicity. So amino acid is considered as an ampholyte because of the presence of carboxylic group. So it's the carboxylic group that make it to actually have acidic nature, while the amino group make it to have a basic nature. So that is why amino acid is considered as an ampholyte. So the answer is option E. So it's because of the acidic nature or a basic nature of an amino acid. That is why it is considered or it function as an ampolite. So let's move to question 6. The formation of a peptide bond between two amino acids is an example of dash 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 reaction. A. Cleavage reaction. B. Condensation reaction. C. Group transfer. D. Ionization. And E. Oxidation reduction. So please, ladies and gentlemen, as I am asking you the question, please write the question and the answer. Write the question and the answer so that at least I will know exactly the questions that you are answering. So the formation of a peptide bond between two amino acids is an example of dash 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 reaction. A. Cleavage. B. Condensation reaction. C. Groove transfer. D. Isomerization, and then E. Oxidation reaction. Please, what is the answer to this question? What is the answer to this question? What is the answer to this question? I am waiting for your response. What is the answer to this question? Is it cleavage? Is it condensation? Is it group transfer? Is it is it isomerization? Is it oxidation? So which one is the answer? Condensation reaction. I'm waiting for your answer. Okay, someone said oxidation reduction. No. Please, what is the answer for this question? Okay, someone says question 6 is E. Question 6 is E. No, it's not E. Question 6 is not E. Please, any other answer for question 6? Any other answer for question 6? Any other answer? Yes, correct. And Zaku, that is Raquel, the answer is B. Yes, the answer is B. Question 6, the answer is B because it is a condensation reaction where one amino acid joined with another amino acid to produce a peptide bond. So as a result of this peptide bond, actually a condensation is actually a condensation reaction because a condensation reaction is a reaction where monomers joined together to produce small molecules. So in condensation reaction, generally a smaller molecules that usually eliminated is either ammonia or water molecules. So if you look at when amino acids or two amino acids join together at the end, you realize that molecules of water are released or are eliminated. So that actually make it as a condensation reaction. Achiever Jasmine, I realize that you are the only one that joined my YouTube community. So thank you very much for doing that. So let's move to question 7 without wasting our time. So question 7. So the answer is condensation. So question 7, which of the following factors is responsible for the denaturation of proteins? A, heat. Sorry, which of the following factors is not responsible for the denaturation of proteins? A, heat. B, charge. C, pH change. D, organic solvent. So ladies and gentlemen, what is the answer to this question 7? Which of the following factors is not responsible for the denaturation of proteins? A. Heat. B. Charge. C. pH change. D. Organic solvent. So what is the answer to question 7? Write the question and the answer. What is the question, please? I'm waiting for your answer. Question 7. Question 7. What is the answer? What is the answer? What is the answer? Okay, 7B. Excellent. Excellent, Abdul Halim Garba. Excellent, Jamal. Excellent, Zulaihat. No, Zulaihat, you are not correct. Okay, excellent. Also, Rachel. So when we said protein, generally proteins has 4 level of structure. We have primary structure of protein, we have secondary structure of protein, we have tertiary structure of protein, and then the quaternary structure of protein. So these are the 4 levels of protein structure. So generally, protein structure can be affected through denaturation. When we say denaturation, it means when the protein loses its three-dimensional structure. So whenever protein loses its three-dimensional structure, that tells you that the protein is actually the functions of the proteins will be lost. The function of the protein will be lost. It can be an enzyme. So an enzyme or a protein can lose its function when it undergoes denaturation. So denaturation is a process where a protein loses its structure, which subsequently lead to the loss of function of the protein, and that is what we call denaturation. And there are so many there are so many factors that can affect the three-dimensional structure of protein. So heat, most of the protein, they function or they work at a particular temperature. So if they exceed that temperature, the protein will lose its structure, because when there is an increase of heat, there is a likelihood the peptide bond will be affected, so the protein will be digested into the individual amino acid, or the hydrogen bonding, the disulfide bonding, van der Waals force of attraction of the proteins will all be affected, and as a result of that, the dimension or the three-dimensional structure of the proteins will be lost. So denaturation is a process whereby the three-dimensional structure of the proteins will be lost. So now, what are the factors that affect the denaturation of a protein, or that lead to the denaturation of protein? So heat is one of it. So whenever you expose your proteins to high temperature, so there is a likelihood all the bonds in the proteins will be affected. Then second, charge. So a charge which can be positive or negative, it doesn't actually lead to protein denaturation. So most of the charges, they are even actually aids in folding, twisting of the protein. So instead of denaturation, it's most of the charges, they actually increase or they stabilize the protein structure. They stabilize the protein structure, not denaturating. So that is why pH, that is why charge doesn't affect the denaturation process. So charge here is the answer. And then pH, and a pH can be above 7.0, it can be below 7.0. So some proteins, they are highly stabilized in acidic pH, while some protein they are highly stabilized in an alkaline pH. So if you have protein that actually stabilized in basic pH and now you take it to acidic environment, so definitely the structure of the proteins will be affected. The structure of the proteins will be affected. So pH change lead to denaturation. Then organic solvent. So some of the proteins, if you take them to an environment where the solvent is not suitable for maintaining the integrity of the proteins, then the three-dimensional structure of the proteins will also be affected. So, ladies and gentlemen, a simple term when you said protein denaturation means it's a process whereby protein loses its three-dimensional structure, which subsequently leads to uh, loss of function. So the answer here is charge. So charge doesn't lead to the loss of three-dimensional structure of protein. So the answer is charge. So let's move to question 8. So in question 8, I already even wrote the answer. The question says, which of the following is responsible for specifying the 3D shape of protein? A, peptide bond. B, the amino acid sequence. C, interaction with other peptides. D, interaction with other chaperones. So, ladies and gentlemen, generally, proteins has its three-dimensional structure. So, the 3D structure of the proteins is maintained due to the amino acid sequence. So, the amino acid sequence of a protein is what will determine how the 3D structure of the protein will be. So, generally, proteins have different structures, they have different shapes, they have different structure. So the shape of the protein varies from one another, and that shape and the structure of the protein, or that different shape of the protein, is due to the amino acid sequence. So it's the amino acid sequence that are present in the protein that will determine how the shape of the proteins will look like. Go and check the shape of hemoglobin. It's not the same with the shape of myoglobin. Go and check the shape of insulin and the shape of adrenaline, the shape of oxytocin, the shape of somatotropin, the shape of some important other enzymes that are found in the body. Their shape are different, and why are they different? Is because of differences in the sequence of their amino acids. So let's move to question 9. Let us move to question 9. Question 9 says dash 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 is not a classified form of conjugated proteins. A. Lipoproteins. B. Glycoproteins. C. Metalloproteins. D. Complete proteins. So, ladies and gentlemen, when we said conjugated proteins or glycoconjugate or conjugated proteins, it means when you have proteins joined with another molecules. So now I want you to answer this question. Question 9. What will be the answer? Dash 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 is not a classified form of conjugated proteins. A. Lipoproteins. B. Glycoproteins. C. Metalloproteins. D. Complete proteins. Excellent, Ibrahim Bashir. The answer is complete proteins. So a complete proteins is not a classification of conjugated protein. So lipoproteins is a conjugated protein. So when we say lipoproteins, it means when you have a combination of proteins and lipids together. Glycoproteins when you have a combination of sugar and proteins, while metalloproteins when you have proteins and metal like hemoglobin. Hemoglobin contain iron, so it's an examples of conjugated proteins. So complete proteins is not an example, or it's not a class of conjugated proteins. So now let's see. So the answer is complete proteins. So let's move to question 10. Which of the following proteins was first sequenced by Frederick Sanger? A. Myosin. B. Insulin. D. myoglobin, and D, hemoglobin. And I already write the answer to this question, which is insulin. So, ladies and gentlemen, actually, Sanger is a scientist that was able to sequence the whole proteins, or that was able to show the steps and the processes of sequencing protein. When you said protein sequencing, it means determining the amino acid that are exactly present in a particular protein. So the answer is insulin. So insulin was the first protein that was actually sequenced by Friedrich Sanger. So let's move to question 11. Question 11. Which of the following statement is true about proteins? A. Proteins are made up of amino acid. B. Proteins are essential for the development of skin, teeth, and bones. C. Proteins is only nutrient that can build, repair, and maintain body tissues. D. All of the above. So, ladies and gentlemen, which of the following is correct? Question 11. Achiever, you said question 11 is B. No. Question 11 is not B. Raquel said question 11 is D. Yes, question 11 is D. The answer is D. That is all of the above. So option A, B, and C are the functions or are true statement of protein. So the answer is D. So let's move to question 12. How many amino acid make of a protein? So we know that there are about 20 amino acids. There are about 20 amino acids that are used in building protein. So we have 20 of them. Yeah, of course, achiever. Proteins they have in for bone and teeth, yes, and skin development. Of course, protein generally is used for growth and development of the body. So you see question 11, there are about 20 amino acids that use in building proteins. So let's now move to uh, question 13. What is the bond between amino acid called? Ionic bond, B acidic bond, C peptide bond, D hydrogen bond. So peptide bond is a type of bond that links one amino acid to another. So the answer is peptide bond for question 13. So a bond that joins one amino acid with another is called peptide bond. So let's move to question 14. Which of the following statement is true about proteins? Proteins are polymers of glucose. B. Proteins are polymers of amino acid. C. Proteins are polymers of peptide bond. D. Proteins are polymers of disulfide bridges. Then the answer is proteins are polymers of amino acids. So let's move to question 15. Which of the following techniques is used to determine the protein structure? A. X-ray crystallography. B. Cryptonics, X-ray vision, and then D. None of the above. So which of the following is used in determining? Which of the following is used in determining or which of the techniques is used in determining the protein structure? So which of them? Because generally we have techniques that we can use to determine the structure of a biomolecule. So we have it. So which of these is used? Excellent, 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 Bashir. Excellent, Garva. The answer to question 15 is A. So, X-ray crystallography is a technique that used in determining the structure of a protein. In fact, not only protein, even nucleic acid. So you can use X-ray crystallography to determine the structure of proteins, to determine the structure of nucleic acid. So you can use X-ray crystallography to do that. So let's move to question 16. Which of the cell organelles is involved in the synthesis, or which of the following organelles is involved in the process of protein synthesis? A. Vesicles. B. Ribosomes. C. Sucrotrophs. D. Mitochondria. So which of the following organelles of the cells is used in protein synthesis? So how do we synthesize protein? Which of these is used in protein synthesis? Achieva says B. Ibrahim Bashir also says B. Okay. Rachel says B. So who have, okay, Jamal also says B, question 16, that is ribosome. Excellent. So generally what happens is that the DNA is in the nucleus, and then the DNA will be converted into RNA, mRNA, that is messenger RNA, and then the messenger RNA in the nucleus will be transported from the nucleus to cytoplasm where the ribosomes is, and then the ribosomes will now use the messenger RNA as a template for protein synthesis. So therefore protein synthesis take place at the ribosomes of the cell. So let's now move to question 17. The 3D structure of proteins can be determined by A. Spectroscopy, B. X-ray crystallography, nuclear magnetic resonance, and D. Option B and C. So you see, we already talked about the X-ray crystallography. So nuclear magnetic resonance, NMR, it is also used in determining the structure of the protein. So ladies and gentlemen, you shouldn't be forgetting all this thing. So what are the techniques, the two major techniques that are used in determining the 3D structure of proteins? We have X-ray crystallography, and then we have nuclear magnetic resonance. So you shouldn't forget about this. You shouldn't forget about this. So the answer is option B and C. Option B and C. They are used in determining the structure of a protein. So then now let's move to question 18. Which of the following is true about enzymes? A. Enzymes are proteins. B. Enzymes are nucleic acid. C. Enzymes are carbohydrate. And then D. Enzymes are DNA molecule. So what is the answer to this question 18? Okay. Garba says 18 is A. Jamal also said A. Rachel also said question 18 is A. And then Zainab Sagir says question 18 is D. Then Rachel says A. Ismail says 18 is A. So the answer is A. So enzymes are proteinous in nature, although some are nucleic acid in nature, but 99.99% of 99.99% of enzymes are protein. 99.9% of them. So then let's move to question 19. Which of the following statement is true about the primary structure of proteins? So please, who can tell me what are the 4 levels of protein structure? Please, I need the answer on the comment section. What are the 4 major levels of protein structure? Please, someone should mention that. I'm waiting for the answer. I need answer to question. Sorry, I said someone should actually tell me what are the levels of protein structures. What are the four levels of protein structure? Okay. Docus Nehemiah says primary. Then apart from primary, what next? What is the other levels of protein structure? Okay, primary, secondary, tertiary, yes, and then quaternary. Excellent. So now let's look at the option. So which of the following statement is true about the primary structure of protein? A, they are helical structure of protein. B, subunit structure of protein. C, three-dimensional structure of the protein. And then D, the sequence of amino acid joined by a peptide bond. So please, what is the answer to this question 19? Which of the following statement is true about the primary structure of proteins? A, helical structure of the protein. B, subunit structure of the protein. C, three-dimensional structure of the protein. D, the sequence of amino acids joined by a peptide bond. So which of the following is true about the primary structure? Yeah. Okay, Ayo said question 19 is D. Jasmine also select D. So is there any other answer for question 19? Yes, Zainab Sagir mentioned that the levels of the protein structures are primary, secondary, tertiary, and quaternary structure. Excellent. You are correct. Then what is the answer for this question 19? Yes, question 19, the answer is D. So the primary structure of proteins determined by the sequence of amino acids joined by a peptide bond. So you are correct. The answer is D for question 19. So let's now move to question 20. The process of protein synthesis takes place in which of the following cell organelles? B. Nucleus. B. Vehicles. C. Cytoplasm. D. Mitochondria. So the process of protein synthesis takes place in which of the following cell organelles? So it actually takes place in the cytoplasm because ribosome is located at the cytoplasm. So it takes place at the cytoplasm. So the answer is C. So then question 21. The first amino acid in a polypeptide chain is A, serine, B, valine, C, alanine, D, methionine. So which of the amino acids that is the first amino acid found in a polypeptide chain? Let me see who can give me answer to this question. The first amino acid that is found in a polypeptide chain. That is the starting amino acid. Ayo says 21 is A. Any other answer? Question 21. No, Rahama says 22. Okay, 20, okay. Garba says 21 is A. Banyo, you said D for which question? So you are all selecting A for 21. So the answer is not A for question 21. The answer is not A. Question 21 is not E. Any other answer? Yes, Banyo. For question 21, the answer is D. So the starting amino acid for every protein in nature, for every naturally occurring protein, the starting amino acid is methionine. And the starting codon, I think for those of you that are doing biochem 219, we'll come to that where we are going to see how methionine serves as the first amino acid in a polypeptide chain. So methionine is the first starting amino acid. So the answer is D. So then let's move to question 22. A dipeptide has A, two amino acid and one peptide bond, B, two amino acid and two peptide bond, C, two amino acid and three peptide bond, D, two amino acid and four peptide bond. So ladies and gentlemen, when we said dipeptide, question 22, a dipeptide. So which one is the answer? A, a dipeptide contain two amino acids, one peptide bond. B, contain two amino acid, two peptide bond. C, two amino acid, three peptide bond. Then D, two amino acid, four peptide bonds. So what is the answer for question 22? Achiever says question 22 is B. So any other contrary answer for question 22? Nehemiah says question 22 is B. Any other answer? Okay. So for question 22, the answer is A. The answer is A. Dipeptide means two amino acids, one peptide bond. Tripeptide means three amino acids, two peptide bond. Tetrapeptide means four amino acids, three peptide bond. So you should understand that. So question 22, A is the answer. A dipeptide contains two amino acids and one peptide bond. So the answer is option A. Okay. Then question 23. Which of the following statement is true regarding quaternary structure of proteins? A, refers to organization and spatial arrangement of amino acid within a polypeptide chain. B, refers to organization and spatial arrangement of proteins within many polypeptide chains. C, both A and B. D, none of the above. So please, what is the answer? What is the answer to question 23? Question 23, what is the answer? Question 23. Achiever says question 23 is C. Any other answer for question 23? So Achiever, you are the only person that joined my YouTube. Yeah, very soon I'm going to start uploading videos for members only. Question 23. None of you answer the questions correctly. None of you answer the question correctly. First, what is quaternary structure of proteins and how do they form? How is the quaternary structure of the protein form? Quaternary structure of proteins is formed as a result of combination of many polypeptide chains. Many polypeptide chains. So in a quaternary structure of protein, it refers to the organization and the spatial arrangement of a proteins with many polypeptide chains. So therefore, the answer to this question 23 is B. It's only primary structure, sorry, it's only tertiary structure of the proteins that organize and form spatial arrangement with amino acid within only one polypeptide chain. So if the spatial arrangement and organization is within only one polypeptide chain, so uh that one is for for a tertiary structure, and but for the quaternary structure, the organization and the spatial arrangement is within many polypeptide chains. That is why option B here is the correct answer, because the organization and the spatial arrangement is made for is made for more than three, or sorry, is made for many polypeptide chain. So when you have many polypeptide chain, is for quaternary structure. Go and look at hemoglobin. Hemoglobin it has four chain, two alpha chain and two beta chain. So hemoglobin it is quaternary structure. That is why it has many polypeptide chain. So the answer for question 3 is B. Okay. Wow. Dina Mina, thank you very much. I just see someone joined the YouTube community. So thank you very much for joining the membership. So I always encourage students to join the membership, because there are some videos that if you are not members, you cannot watch them. If you are not members, cannot watch them. You can't see them. Yeah, especially those that are not for Barricam 219, Barricam 211, Chem 2, sorry, Chem 201, Chem 221, Barricam 217, Barricam 213. If you are a member, there are some videos that you cannot watch them. So so now let's actually move to question 24. Says which types of bonding is responsible for stabilizing the secondary structure of proteins? A. Disulfide bridges between cysteine residues. B. Bonding between carbonyl carbon and amino groups of peptide bonds. B. Peptide bonds between amino acids. D. Salt bridges between charge side chains. So, ladies and gentlemen, what is the answer to question 24? Question 24. Someone said B. The peptide bond between amino acid is only considered for primary structure of protein. So in addition to primary structure, in addition to peptide bonds in the primary structure, so what other bond stabilize the secondary structure of a proteins? So you see, Mariama says 24 C, so it's wrong, and then says question 24 is B. So do you agree with Cute? She said that question B. Do you agree with her? So for 24, the correct answer is B. So hydrogen bond between carbonyl carbon and amino groups peptide bond is what stabilize the secondary structure of proteins. You see there, apart from the peptide bond is a covalent bond. Sorry, it's a covalent force. So there are non-covalent forces that stabilize the secondary structure, tertiary structure, and quaternary structure of a protein. So for 24, the answer is B. So it's the hydrogen bonding of the carbonyl carbon and the amino groups of a peptide bond or amide bond. So let's now move to question 25. We are now moving to question 25. So the answer is B. That is hydrogen bonding between the carbonyl carbon and amino groups of peptide bond. Then question 25. A coiled peptide chain held in place by hydrogen bonding between peptide bonds in the same chain is A. Primary structure, B. Alpha helix, C. Beta flated sheet, D. Tertiary structure. So what is the answer for this? Question 25. What is the answer for question 25? Please, I need answer for question 25. Answer for question 25. I need answer for question 25. 25B. Mariama says D. Jamal says B. Optimistic says B. So any other answer? Let me see more response, then I will give you the correct answer. So generally, ladies and gentlemen, the secondary structure of proteins is divided into two. There is alpha helix and beta flated sheets. So the alpha flated sheets is the types of the secondary structure of protein that held in place by hydrogen bonding between a peptide bond in the same chain. Remember, the peptide bonds in the same chain. So the answer to this is alpha helix. So when you have hydrogen bonding between peptides in the same chain, that is actually what describe the alpha helix. So the answer to this question is alpha helix. But for the beta flated sheets, let's look at this. A structure that has hydrogen bonding between polypeptide chain. Hydrogen bonding between polypeptide chain arranged side by side is A. Primary structure, B. Beta helix, C. Alpha flated sheet, D. Tertiary structure. So what is the answer to this question? What is the answer to question 26? I need answer for question 26. Okay, 21. Murjanatu says C. Jamal says C. Yes, the answer is C. So when you have hydrogen bonding between polypeptide chains, so different polypeptide chains, when you have hydrogen bonding between different polypeptide chains arranged side by side, that is for beta flated sheet? So the answer to this is beta flated sheet. So let's move to question 27. In alpha helix, the bonding is between the A adjacent amino acid, B carboxylic carbonyl oxygen of one polypeptide bond to the amino group of the fourth acid away, D carbonyl oxygen for second peptide bonds to the NH groups of the third amino acid away. Then D carbonyl oxygen of second peptide bonds to the amino group of the fifth amino acid away. So the answer is B. So the bonding is formed between the carbonyl oxygen of one peptide bond to the NH, that is amino group of the fourth amino acid away. So it means that the bonding is between oxygen of one polypeptide, of one peptide bond, and then joined or linked with the amino group of the put amino acid away. So the answer is B. So let's look at question 28. The primary structure is primarily maintained by A peptide bond, B hydrogen bond, C ionic bond, D hydrophobic bond. What is the answer for question 28? I need answer for question 28. Please, what is the answer for question 28? No answer. Question 28. Primary structure is primarily maintained by peptide bond. Excellent, excellent, Murjana to Sagir. So, how do I become a member? Yes, to become a member, you can check my YouTube. You can see join. So, you click on join, then you use your ATM card to place or to put your details. Then 750 naira will be deducted from your account. So once you are a member, you will have access to all my videos on the channel. So the answer is peptide bond for 28. So ladies and gentlemen, I just wanted to tell you, for most of your courses like biological techniques, bio 213 will do similar. For biochem 217 will do the same. For biochem 213 will do the same. For biochem 211 will continue. So we are not actually for this 211. Maybe we are going to have another interaction on Friday or Thursday. Are you happy with this kind of interaction? So that at least we'll be getting first question paper. And so the answer, so are you happy with this, so that at least we'll be taking our time getting past Q and solving them here? Are you happy with that? Okay, Fatima Mustafa says yes. Okay. Let me tell you, sometimes if you are not even a member, if you are not a member to the YouTube, you cannot even write on this comment section. So YouTube used to do that. So YouTube used to do that. If you are not a member, sometimes it will not even allow you to write on the chat box. So that is why I will advise you, if possible, you try and join as a member. It's not expensive. So it's the YouTube policy that if you want to become a member, then you register as a member, which will allow you to have access to all content, to have access to all YouTubers. So let's move to question 29. So the answer here is peptide bond. So 29, what is the pKa range of the carboxylic group in alanine? A 3.5 to 4.0, B 8.8 to 11.0, C 5.0 to 7.0, then D 1.8 to 2.4. So the answer is 1.8 to 2.4 because alanine is an amino acid that can that can actually that has a pKa range of about about 1.8 to 2.4. So let's move to question 30. Amino acid valine exists as dash 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 at neutral pH. A. Cations. B. Anions. C. Neutral. D. Zwitter ions. So at neutral pH, what is the nature of valine? Is the valine cations? Is it an anionic state? Is it at a neutral state? Or is it at Zwitter ions? So what is the answer to question 30? Question 30. Is it Zwitter ion, cation, anions, or neutral? If you're answering the questions, you please write the question number and then the answer. What is the answer for question 30? No, question 30 is not Zwitter ion. It's not Zwitter ion. So the answer is neutral. So valine is neutral at neutral pH because Zwitter ion and neutrality of any substance are two different things. When you said it is neutral, it means that it doesn't have positive charge, doesn't have negative charge. But for the Zwitter ion, they are charged. The amino group has positive charge, the carboxylic group has negative charge, but the charges neutralize each other, and that is why they are considered as Zwitter ions. They have charge, but they still maintain zero charge at the end. They all maintain, so the amino acids maintain zero charge because the negative charge and the positive charge from the amino group and that of the carboxylic group, they will now nullify or neutralize each other. So now question 31. So the answer is neutral. So Zwitter ion dash 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 at neutral pH. A uncharged, B positively charged, C negatively charged, D charged but net charge is zero. But net charge is zero. So what is the answer for question 31? What is Zwitter ion? Excellent. So Muhammad Farouk, the answer is D. That one is D. They are charged, but their net charge is zero because they have negative charge for carboxylic group and positive charge for amino group. Okay, so excellent. You see, yeah, excellent. Ayuba, Mariam, Jamal, and Farouk. The answer is D. So they are charged, but they are charged and neutralized, making them overall to have zero charge. Excellent. So the answer is D. So 32. The titration curve of glycine represents A, 2 buffering region, 2 sorry B, 2 pKa value, C, both A and B, D, 1 buffering region at neutral pH. Please, what is the answer for question 32? Yes, Rookie Adam also selected D for that one. So for 32, what is the answer? C, both A and B? No, no, Bashir. 32. Excellent. 32 is B. So most of the amino acid, especially those amino acids that have the ionic form by both carboxylic group and amino group, their pKa values are usually 2. They have 2 pKa value. And if you want to calculate the pi, that is isoelectric point of the amino acid, then you can take the 2 pKa value and divide them by 2. So you take the two values and you divide them by 2. So ladies and gentlemen, we are going to stop here, and I think we have actually discussed and we look at some of the questions. So what I can tell you, you can still go back. This video will be on my YouTube channel, so you can still check it from beginning to the end, so that you will have an idea of all the questions that we have solved for those of you that joined late. So ladies and gentlemen, I hope you are happy with the interaction. And when do you think we should still go back to this course before you write the test? So that at least we'll go into the material and pick other questions. All these questions that you can see here, I picked them from the material. I've picked them from the material. So when do you think will be the best to continue with this interaction before you write the test? And I would encourage those of you that actually didn't join the YouTube membership to try to do that as much as possible, as soon as possible, so that at least you will not be left behind. Okay, Mariama says a day before the test. When is the day before the test? Okay, on Sunday. Okay, so which day are you writing? Okay, your effort is fully appreciated. Okay, you are writing the test on Monday? Is it on Monday you are writing the test? And let me say this, if you know that you are writing the test, okay, yes, it is on Monday. So if the test is on Monday, someone is writing on Wednesday. Please be sure, when are you writing this test? Amen Murjana Sagira, thank you very much. Maria Musa says, you too fast. Am I not? I'm not fast. I think we followed the question very gradually. Or let me ask this question. Do you want me to go slower as this? Do you want me to be to slow to be slow in actually asking the questions and also respond from you? Am I fast? Am I fast? That is what Mariam said. Am I very fast? Please let me just see your response so that at least by next time we'll actually try to see how we can. Which Tuesday? Is it this Tuesday or next week Tuesday? Is it this Tuesday or next week Tuesday? Because tomorrow is Tuesday. Okay, next week Tuesday. Okay, we end it now. So please, in case if you want to still revisit what we have discussed, so we are writing the test next week Tuesday. So Monday will be okay. Okay, I'm not fast. Okay, sir, it is okay. Okay, so but why not to make it weekend? Because Monday is a busy day, so I might not probably have enough time. So I think maybe we can take it to we can do it on Tuesday, I think. Sorry, on Sunday, either Sunday or Saturday. Because Monday is too close. It's too close. It's better you actually after reading, like from now to Sunday, you maybe have gone through a lot, and then you now come back and look at the questions. So we look at the questions here, then you have maybe one day to read again. So thank you very much, Jasmine. I am happy that you joined the YouTube membership. Dina Maina also, you are welcome, and I'm happy that you joined the YouTube membership. So please, for those that didn't join, I encourage you to try to join so that at least you'll have access to all the videos that we are going to upload in the website. So thank you very much, and ladies and gentlemen, bye bye. See you next time. So I'm going to end the meeting. Till next time.